Good afternoon, everyone. Welcome to our Immuno-Oncology webinar. My name is Emma, and on behalf of Oxford Global, I would like to thank you all for joining us today. Today we have Ivan Plavec, Chief Business Officer at ImaginAb, who will be presenting on how CD8 imaging is useful to pharma regarding drug research and clinical decision-making. After the presentation, we will have a 5-minute question and answer session. Please submit your questions through the chat option on the bottom of your menu display. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. Without any more delays, I would like to get started with Ivan's presentation. Ivan, over to you.
So welcome everyone to today's seminar. I'll be talking about the use of CD8 imaging in the space of immunotherapy and how this imaging is useful to pharma regarding drug research, but also touch a base upon the work we've done on interviewing clinicians to understand how CD8 imaging would be used in clinical practice to advance the health of the patients and improve the use of immunotherapies. Now, since ImaginAb is the only company that at the moment has a CD8 imaging agent in clinic, and by default, I'll be talking a little bit more about ImaginAb's projects. But in the first part of the talk, I also give a general points as to why such imaging would be important and where the field is in thinking about imaging in immunotherapy. So why it is important to image immune cells in immunotherapy? I think for some of you this might be obvious, for others maybe not so much. So I think it's worth spelling out that immunotherapy and specifically checkpoint inhibitors, they have truly transformed the landscape of cancer care. Cancers like melanoma used to be a death sentence 2 decades ago, and now there is a great success in managing this disease. Now, checkpoint inhibitors have been on the market now for several years, and suffice to say that there is a large variability in patients' responses. And it's not surprising if one thinks about immunology and variability in immunology among individuals. So there is a need and great understanding that personalized assessment of immune cell activity in individual patients has the potential to greatly improve the effectiveness of immunotherapies. And so we and other people have thought about this, and ImaginAb specifically is developing a pipeline of PET tracers that are specific for immune cells. And our lead product is a tracer to image CD8 T cells. And so why CD8 T cells as opposed to some other cell type? It's primarily driven by the work of others. This is an image from a paper coming from Tony Riba's group a few years ago in melanoma patients, looking at the biopsies and realizing that there is a direct correlation between CD8 T cell counts in the tumor upon treatment, as a predictor of response. And it's not surprising. These are cytotoxic cells. They are a key player in killing a tumor, but it is one of the situations where a hypothesis, immunological hypothesis, is fairly straightforward. The more cytotoxic T cells in tumor, a better outcome for a patient. Now, of course, there are the questions about, are they activated, are they exhausted, energized? These are, if you wish, second questions that the current field, at least in clinical practice, the message is fairly clear. You just need to drive these T cells into a tumor. So having a good measure of the T cell count in a tumor is an important element in the a toolbox that the clinician can have to treat patients. So you could address this question various ways. If you think about the patient, you can see here on the left-hand side is a diagram of a cancer patient that's receiving IO therapy. These are generally later stage patients with metastatic disease, having multiple lesions throughout the body. So you could, envisage taking a blood sample and measuring certain markers in a circulation, like circulating tumor DNA, maybe cytokines, maybe, analyze immune cells by fax. But by and large, it gives you an average response. The fact that the lesions in the body heterogeneous is not captured well by looking at blood samples. The way it's done right now in clinic, if you're diagnosed, you go to hospital, you get your biopsy taken, and then one can analyze the biopsy sample, which is a common practice, and it's very useful for staging patients. But again, you're looking at the single lesion. Many lesions, especially in the lung cancer, the location of a lesion is as important as the size of the lesion and the nature of it. So it is a snapshot. A biopsy really gives you a snapshot, and hasn't been that useful for clinicians to make decisions about the immune response. They're great to stage a cancer and get some initial understanding. There's also just a procedural issues. Biopsy is a minor surgery. Many patients don't like to do it, and then even in the cases where you are collecting a biopsy, it's a hit and miss. Generally, we hear from clinicians about half of the biopsies fail because you just didn't get the useful tissue. So there are limitations there. Where we're focusing on is imaging, and that's all the way on the right-hand side. So the imaging allows you to see the whole body. It's non-invasive, right? So you just it's a simple procedure of infusing a tracer, and then patient sits in a PET machine. It's easy to do, and you are looking at the whole body image, and it jives really well with the clinical practice, because clinicians are imaging cancer patients anyhow. They're doing CTs and MRIs for diagnostic purposes, also to monitor the RECIST criteria of a patient to see how they're progressing on treatment. And so imaging, it has a natural place in that particular therapeutic field. And so what also imaging does, specifically when we talk about imaging immune cells, you can look at all the lesions at the same time, see what kind of cells are there in those lesions, but not only tumor lesions, but also throughout the body, looking at normal and draining lymph nodes, looking at normal hematopoietic organs, to get a much better understanding of where the cells are moving, how they're changing. And that has never been done before, and is what we are trying to achieve with our tracers. So here on slide 5, I just listed the different techniques one could use to image immune cells. Obviously, one can use various sort of metabolic probes, fluorodeoxyglucose, or more commonly known as FDG, is commonly used to stage patients. It measures metabolic rate of a tumor, or I should say, measures the metabolic rate of a cell. Tumors tends to be more metabolically active, so they light up. Small molecules, not very specific, not very easy to specifically visualize an immune cell. One could do pre-labeling of the cells with various particles. One could introduce reporter genes, but that requires an ex vivo manipulation, which in many cases is just not possible. And if you think about immune cells, they are generally defined by antibodies, staining with antibodies, and by their surface markers. So this is a mainstay of immunology to understand and classify cells, is by staining. And so using antibodies or minibodies or nanobodies, so protein-based, more specific tools, is really the best choice for immunology. Now, of course, there are a number of different strategies you could take to develop an imaging probe. And I'm kind of putting this in a context here to understand why we focused on minibodies and diabodies. It has a lot to do with a sort of a Goldilocks phenomena, if you wish, that you need for an imaging agent. You need something that's specific, that enters the body quickly. You don't need the high dose, so it can be safe. And then it should clear also really quickly. It should be non-immunogenic and inert, and have a great specificity, such that when you look at the image, you have confidence that what you're looking at are actually cells and not some background issue. So, antibodies historically, we know, it's very easy to achieve high selectivity and affinity, and it's easy to make probes. Nowadays, techniques, have so advanced that you can readily make antibodies against any epitope, virtually. But they have their problems. They're not biologically inert. And they can be immunogenic. Of course, they can cause ADC response or maybe complement, directed cell killing. So there are some issues with them. Maybe the biggest issue with full-length antibodies is that their PK is just way too slow. So it takes maybe 4 or 5 days of clearance before you can actually image a patient. So they're not really practical from that perspective. On the other end of the spectrum, you have small molecules. They're really not that specific, and actually it's not that easy to generate a probe, to generate really a good small molecule probe. Peptides, similarly, specificity is an issue. They clear maybe too fast, and stability in vivo is also an issue. But then again, in each of these classes, there are success stories, but none of these offer a kind of a stable platform where you could readily develop probes. And then we enter into this sort of a small molecule, small protein binders, the single domain variable fragments. There's kind of proteins, aptamers, camelids, small fragments that will have a good affinity, which is not always easy to achieve, but yes, you can do that. But then again, they tend to be immunogenic in many cases, and so repeat administration may be problematic. Where we focus on is minibodies and cis diabodies, because as you can see here, is mostly most of green dots are here, and especially minibodies where we've done most of the work. Super easy to make. Achieving selectivity and affinity is as straightforward as building an antibody. So work that we've done here is the 2 bottom green dots, which is make them biologically inert and non-immunogenic, and we have now multiple examples that this is so. And the most important is the PK and biodistribution that's suitable for imaging. And I'll show you this on the next slide. If you look at the PK properties of proteins of different sizes, you see that minibodies and cis diabodies fall into that middle range, which is, I mentioned the Goldilocks, not too hot, not too cold. So they enter the bloodstream really rapidly, and they clear rapidly, but not too fast, such that you couldn't get the good images. So we can see images as early as 1 hour after infusion, where with minibodies, where for the intact antibody you may have to wait 4 or 5 days. So clearly. So to introduce minibodies, they're bivalent. In contrast to sort of single-chain variable fragments and many other smaller proteins. They retain specificity and affinity of the parent antibody fairly readily. They have a half-life reduction, so they clear from the body really fast, and because they don't have Fc region, a binding portion of the heavy chain, there's no non-specific binding. And clearance is faster. They're non-immunogenic. We've tested this now in clinic. There are no effector functions, so at least the minibodies that we've done so far, they have no impact on cell activity, either positive or negative. They do clear through kidneys and liver minibody, CD8 minibody predominantly through liver, which is also a good feature because the rest of the organs, when you see metastasis, commonly are clean. And you can conjugate various types of imaging moieties and minibodies, fluorescent labels, radioactive proteins if you want to do therapeutics, or various types of imaging radioisotopes. Which leads me to a tracer that I'm going to be talking about today in the second part of the talk, which is simply named 89 zirconium desferoxamine IAB22M2C, which is a mouthful. So I'm going to refer to this as a CD8 tracer for the rest of the talk. It is a minibody. You see on the left-hand side the structure of the product is a minibody that has a desferoxamine chelator covalently attached. There's about 3 molecules of desferoxamine per minibody, and then you capture zirconium just by virtue of having a chelator. That is a product that is infused into a patient. Because zirconium has a half-life of 78.4 hours, which is about 3.2 days. The product has a reasonably long shelf life for a nuclear medicine product. It's about 6 days, within which we can centrally manufacture and deliver to hospitals at present for our clinical trials and clinical trials of our partners. Affinity is good, 0.4 nanomolar. It's small size, it clears fast. It is immunologically inert. We've tested this in many different ways, and now in clinic we haven't seen any immunologically related responses, such as anti-drug antibodies, cytokine response, any of that. And we've shown that there are no effector functions within that product. Now we've tested, of course, preclinically, and here is an example of an animal model where you can see on the left-hand side it says a non-responder. This is a tumor in a mouse, and if you do the cross-section of the image, you can actually see this sort of an immune-excluded tumor where there's a ring of staining around the mass of the tumor, and then in animals that respond to, in this case, anti-PD-1 antibodies, you see that the tumor has now been infiltrated by CD8 T cells, and if you do the math, you measure the intensity of this staining, then you see about a 3-fold increase in the responder. Now, one thing I forgot to mention is if you're not familiar with PET as a technique, it is actually a quantitative technique, right, as opposed to MRI or CT scan, because the tracer is in the tumor. The density of the tracer that's retained in the tumor, it's releasing the radioactive, during the radioactive decay, you collect the counts. So it's a very analogous to, many other radioactivity-based techniques where you can actually use it for quantitation. And that's, of course, important element. That's why we're using PET. So with that introduction, I'll switch to our clinical data. We have completed the phase 1 study last year. I'll present this, and we are running currently a phase 2 study. The data were presented at SITC last year by Michael Gordon, who was our lead investigator, and so I'm borrowing some of his slides to show here. The study was very simple. It was meant to address safety, tolerability, and whole body distribution of a tracer, determine optimal dose. So we did the dose escalation arm of the study, and then the optimal scanning parameter. And so you see here, we would infuse a tracer, 3 millicuries of it, and then at different doses, and then a patient would be brought into a PET center to take images, sometimes as early as 1 to 2 hours after infusion, and it can be as late as 7 days. We actually had one patient at 10 days. All these patients were cancer patients with solid malignancies, at least one RECIST measurable lesion on CT or MRI, and they were some treatment naive, some on the checkpoint inhibitors. And here's the study demographics. You actually see here on the bottom all our 15 subjects, their images, and you know, with some variability in terms of clearance. So you can see sometimes a little bit more liver or kidneys, sometimes more in the large intestine where the primary site of clearance is. The images are strikingly similar between the patients overall, right? So we had some melanoma patients, 8 of them, 6 lung cancer patients, and 1 hepatocellular carcinoma patient. Some were treatment naive, some had prior CPI treatment, but not at the time we imaged them. And most patients actually were on the treatment at the time when we imaged them. And here is the first image I'll walk you through to get the idea of what this looks like. This was a liver cancer patient that was on nivolumab for 2 weeks prior to scan. You can see here day 1, it's a 1-hour scan, and on the right hand side the 4th image is day 6, which is a 6-day scan. So you see that even at 1 hour we see essentially the same structures as we see at day 6. So very stable signal, and what immediately becomes apparent is that what we are seeing are the sites of a normal lymphoid organs in the body. So we see spleen. There's a blue arrow pointing at the spleen. We see bone marrow, so you see the outlines of the pelvic region, you see the spine, you see the ribs, and some of the large bones. And then when we look at 24 hours, you start seeing lymph nodes, and of course we do see tumors. These green arrows pointing to the liver cancer region in this patient. I'll show you some other patients, you know, that don't have liver metastasis and the liver is clean. So no background in the bladder, no background in the colon, no background in the pancreas, lung, brain region, large muscles, heart. So the areas where one mostly worries about metastasis, they're all clean. So it's a very as imaging agents go, it's a strikingly clean image and a signal. We see on the right hand side is the cross section through the body, pointing specifically at liver, and on the top you see the hepatic phase CT scan and the lesion that's visible on the CT scan. When we look at the staining for the CD8 PET, which is on the bottom, it's actually, the way this is done is you do PET CT scan. So there is a low intensity CT just to get the anatomical features, and then the CD8 is shown here in the false coloration as red on the red scale. And you see here actually 2 lesions, one that coincides with the lesion that's seen on the CT scan, and the new one that wasn't so easily visible on the CT and actually was missed by the first pass analysis. And when the clinicians went back and looked at it, and there was actually a occluded lesion there as well. So showing that the CD8 PET scan was actually even maybe a better tool for staging patients than a CT. Now, of course, you can't expect that the T cells are always going to be in a tumor, so it's a little more complex story here. But the combination of the CT and the PET gives a very good understanding of the patient's tumor location and the presence of CD8 T cells. So we were very excited. This was our patient number 2, and so it essentially, you know, was, the picture says 1,000 words, was the first time that anybody has actually seen the biodistribution of CD8 T cells in the body. And we then continued, collected more patients, and of course we're looking at the variety of situations. Here's another patient where they had very large FDG PET avid lesions, and you can see here on the left, which says FDG PET, it's a lung cancer patient. You see in the middle panel these lesions about 7 centimeters across, I believe, and then there's a neck lesion and one in the liver. And when we look for the CD8 PET staining in that particular patient, we haven't seen anything. So these lesions are immune deserts. And that was very important for us because with an imaging agent, you always worry about this EPR effect, which is essentially, in layman's term, means that a probe hangs around in a tissue and doesn't clear just because of non-specific or some kind of retention effect. And we don't see that at all. And so the clearance is excellent and everything. So when you do the time activity curves, you see actually even in this patient, the probe moving into a tissue and then clearing out within 8 to 24 hours entirely. And then you just get the background, which is very low to start with. So confirmation, even though for patient not the great news, but I think for our technology, it was an important piece of information. Now here is one situation where things getting more complicated. So this was a lung cancer patient, and on the diagnostic CT on the left, you can see in the green square where we show where the lesion is. And then you can see I think these are the hilar nodes over here, pointing by the green arrow. And then this patient was on Keytruda for a month, and you can see a very high activation in the hilar node. The sort of the SUV, which is the measure of the amount of radioactivity in AX, here is 7.1, and for the tumor is 2.1. So above background, but also heterogeneous staining. You can see that half of the tumor actually there is no T cell at all. So these are, this is very much consistent with the literature where people have done resections and look for the CD8 distribution by immunohistochemistry, and by no means every tumor the CD8 T cell distribution is uniform throughout the tumor. So it is something that we would have expected, and it shows the technology can deliver that level of resolution. And then, of course, we have 15 patients. I'm not going to, go through all of them. Just point out some features here. This is a late-stage metastatic melanoma patient, and you can see here on the top 2 panels on the left-hand side the adrenal metastasis that has quite a good staining for the CD8 T cells. And then in the middle, you see a muscle metastasis that's very large on the CT scan. And when you look at the CD8 staining, there's a shadow staining that's actually like a ring around the metastasis when you do this more careful analysis. So it is an immune excluded tumor and relatively poorly infiltrated by T cells. Now, because we also see normal immune organs, the bottom down, you see a lymph node, peritoneal lymph node, that's merely 2 millimeters across, and we can see it very nicely on the scan. And you can see on the right-hand side the different view of the patient with those tiny lymph nodes in the peritoneum that are activated because of the surgery that this patient underwent and had the lymphedema. These are not tumor impacted nodes, but they're actually just an immune activation. So very interesting sets of observation coming even just from phase 1. And this is the last slide I'm going to show to demonstrate that we can actually see also brain metastatic lesions by CD8 PET staining, and that is important. You can see here the CT scan on the top actually didn't reveal anything, and the clinicians first missed it entirely. When our team analyzed the CD8 PET images, we observed the site of accumulation that became very strong at 2 and 6 days. Asked the doctor to go back, and they did an MRI scan, and you can see in the fused PET MRI image that the staining is actually within that lesion. So looking at the body whole, we've, even in this just phase 1, 15 patients, you were able to demonstrate the ability to measure the CD8 T cells, both in terms of their density, if you wish, by the SUV max, and as well as distribution by the overall image quality, to see the profiles. Closing on the phase 1 trial results, it is a very safe technology, so we don't have seen any adverse events. There's a robust tracer uptake up to 7 days post-injection, high background to noise ratio, and everything looks very, very good. We picked the appropriate dose, which ended up being the microdose from FDA perspective, so adding additional level of safety to the process. So with these data, we then went on to start a phase 2, and in phase 2 study, we are looking at multiple injections of a tracer to see the profile before and after immunotherapy. It's an ongoing study. We have 11 subjects enrolled to date. We again haven't seen any drug-related adverse events, excellent image quality as in phase 1, and we are starting to see the proof of concept, namely that upon treatment, some patients that respond, we can see more T cells in a tumor, and so we are working through this process to make this, going from proof of concept to actually making this eventually a tool that will be approved by the FDA. See if this works. Yes, I hope you can see the movie that's playing here. So, just to impress upon you one more time, these are the first ever images of CD8 T cells in a human body. These are, of course, 3-dimensional images that provide a high level of resolution, a very, very clean tracer. So as tracers go, I think we were extremely pleased with the performance in phase 1. Now, not only us, the purpose of this talk is to give you also some idea how this is now being used by others. And Pharma has we've been like looking at us as we were completing our phase 1, and as the data came out, said, okay, it's time to use this technology. And we announced several deals. We closed deals with Merck last year, then came Nectar, and then more recently we announced a deal with Roche. And these are all a license agreement where we enable our partners to use the CD8 imaging in the clinic. So why would they do this, right? Why would Pharma want to use this in the clinic? And it's pretty much straightforward. I think the investment into IO therapy is very large. The ability to compare to standard of care checkpoint inhibitors is limited. There are really no pharmacodynamic markers. And so the first and foremost interest from Pharma into this imaging is to use CD8 as a pharmacodynamic biomarker for new IO therapies. Of course, to demonstrate mechanism of action, because most of them are actually supposed to drive T cells into a tumor. And of course, the more longer terms is to correlate to clinical outcomes, and then use the CD8 imaging to better design the pivotal studies, of course, to improve economics with a better design, as well as to have a more rich data set to improve the success of the pivotal studies for therapeutics. And in general, these study designs kind of along the lines as it's shown down here in this image. So patients that are eligible, they would come and get infused with the CD8 tracer, and we would take a baseline scan just to see what is the situation before therapy. Then people go on therapy, and then we will take an on-therapy scan, maybe 3 to 4 weeks into a therapy, maybe shorter, depends on the treatment regime for the therapeutic modality. And then patients are followed just by conventional scans, and they either progress on the disease or they're responding or have a stable disease. So then one can develop the statistics. And this kind of use is the primary use for the pharma. Now we have also, as we completed phase 1, interviewed a lot of clinicians, including payers, in the US and Europe, to understand how would they, feel about this technology? Do they find it valuable? And if they do, how would they apply this in the clinic? So I think from the clinician perspective, there are different questions they worry about, right? And so we were, the response was extremely positive from the clinical community, and where they felt was the most important thing was the scans number 1 and 2, that is very similar to our phase 2 study. So you want to scan before you give a patient a therapy to establish eligibility, right, for a response, non-response, and that may kind of guide you whether you want to do chemo or IO or some combination of the two. And then you scan shortly after you started the therapy to actually see if it is working and how it is working. So this sort of the 1-2 step is where we got uniform response from treating clinicians into the value of this technology. Of course, depending on how the scan number 2 looks like, you either confirm that you want to continue therapy, or you may need to escalate if the things are not working. And then the third application is in patients that have been on therapy for some period of time, and there may be some questions about whether the disease is progressing, whether it's stable, whether the therapy is still actually active. In other words, if I'm giving immunotherapy, are the patients actually having any immune cells in the tumor? And so this is where we are kind of working with the FDA to get the approval ultimately. Now, if you bring this back to the sort of the basics and how we see this moving forward and being useful for pharma as well as ultimately for patients, which is our ultimate goal, is the use of this technology to guide treatment decisions. And so if you look at here, there's sort of a 3 situations that we see. On the top, these are actually real images from our studies. You can see a strong signal. This is this liver cancer patients. It's like uniform distribution of the signal. You see such a weak signal, which is immune excluded areas. You see the T cells around the tumor, but not in the body of the tumor. And then you have cases where there just isn't much of a signal to start with. We are doing IHC in our phase 2, and so we are confirming all these things, the data already confirming it, so no surprises there. But the interpretation here is that you have an inflamed, hot tumor, so you should continue, therapy is working, and keep doing the good job. If you get a weak signal, such as an immune exclusion and a heterogeneous staining throughout the tumor, you might consider adding other treatments that alter tumor microenvironment. Maybe partial radiation, maybe other ways of activating the immune system to get in the tumor beyond checkpoint inhibitors. And if there's no signal whatsoever, you might actually think about some ways of vaccinating the tumor to activate T cells, or breaking the immune resistance in some other ways. And of course, because you're looking at whole body and all lesions, we believe that in the long run, the CD8 PET imaging could actually greatly reduce need for biopsies, especially when patients are on treatment already. Very rarely biopsies are done in the first place. But in some ways, the CD8 PET provides same but even enhanced information relative to biopsies. And of course, we are starting to see the responses of the overall immune system in patients, which should enable us to look both at the immune-related adverse events that people are talking much more about, especially as the combinations come out. So with one picture, you get all the answers, and that's where we think the power of imaging comes into it. And CD8 Tracer is the first one that's really coming out of the woodworks from imaging companies like ours that should add a great value to this fight against cancer that we have. So with this, I would like to stop and invite questions. So this is my thank you slide. These slides will become available to you. Just send me an email, and I think the organizers are going to give you the email as well. There are a couple of slides in the back about ImaginAb as a company, and so how to talk to us if you're interested to do something with us. But I think for the purpose of this seminar, let's just stick to science and I'd like to address any questions from the listeners. And thank you for listening.
Thank you very much, Ivan, for a fantastic presentation. Now, I would like to open the floor for discussion. Remember to send us your questions using the chat feature of the platform. So the first question that we have is, how can Pharma use ImaginAb's CD8 tracer in their clinical trials before it is approved by FDA?
So it's actually very simple. It's not uncommon for imaging tracers to be used in clinic even before they're approved. It's been done before in the CNS space. Essentially, there is a manufacturing file that sits with the FDA that a pharma is using to reference when they use our tracer. And the reason why it can be used before it's approved, because it's extremely safe. The doses that are delivered are very small. It's usually just 1 or 2 injections. And as long as you're controlling manufacturing, everything is fine. So microdose, safe, biologically inert product that clears fast is something FDA is comfortable with.
That's great. Thank you very much, Ivan. We have a second question here, which is, where do you manufacture the CD8 tracer?
So we manufacture in the US at Memorial Sloan Kettering, and we are opening up another commercial site in the US. We do have sites, manufacturing sites that are coming online in Europe and in Australia. So we'll be imaging the first patients in Europe maybe this month. And then the tracer manufacturing will be available beginning of next year, and then about the same timeline for Australia. We're not yet in Asia. It's a longer process, so we haven't yet embarked on that.
Perfect.
So Europe, US, and Australia.
That's great. So we have one more question here. If there's any other questions, then please don't hesitate to send them to us using the chat feature. The final question that we have is, can one use the CD8 tracer in clinical trials in the EU? What about the rest of the world also?
Yeah, so yes, one can use it in the EU and as well as the rest of the world, yes. Qualifying being is that because of the 6-day shelf life of the product, we can manufacture centrally and deliver. We have done clinical trials in Japan in the past, even though we don't manufacture in Japan. So I would say logistically we can deliver anywhere in the world. It's just a matter of whether the country allows import of radioactive tracer, going through the regulatory and administrative process of getting it there. But the easiest thing is to do things in the US and Europe at the moment.
That's great. Thank you so much, Ivan. I believe that's all the questions that we have today, which brings the Q&A to a close. Thank you very much for all of the questions. And once again, thank you to Ivan for this excellent presentation. As he has mentioned, please do get in touch with Ivan for any further information following the webinar. This webinar is part of the run-up to our second annual Advances in Immuno-Oncology USA Congress, which will be taking place on the 8th to 9th of October this year in San Diego. If you would like further information on the Congress or would like a copy of the conference program, please contact the marketing department or visit the conference website. On behalf of Oxford Global, I would like to thank everybody for joining us today. I would like to say a big thank you once again to Ivan, who has put a lot of time into making sure that this webinar was as interesting and valuable as possible. All that remains to be said is have a great day, and we look forward to welcoming you to one of our future events.